what I want to do is to sort of fill in the gap between the kind of microscopy that we just heard about from Ken and what Lee Peachy talked about. That is, what Ken was discussing was a type of microscopy ideally suited for doing ultra-high resolution, essentially atomic structure resolution of what amounts to monomolecular films, of monomolecular crystalline films. So looking at structures that are, that are in the neighborhood of, there is a grid, very thin films that are in the neighborhood of, say, 50 to 100 angstroms thick. Okay. What I'm going to be talking about is techniques for looking at intermediate levels of structural resolution, for looking at structures that range up to a neighborhood of a half micron thick, and looking for internal structures within the specimen. Okay? So the kinds of things that we heard about from Lee was looking at either using stereo microscopic methods to be able to follow either like tubercular lattices or following points on a surface to determine a surface contour. What I'm going to be talking about are techniques that allow you to look at internal structure within a specimen, much the way the high-resolution crystallographic methods that we just heard about allow one to look at internal structure within a single molecule. Okay, first slide, please. Okay, to orient you in sort of what our goals are, all the technology that we've been developing in this area has been developed really to tackle a particular problem in cell biology. And what our laboratory has been interested in, and when I say our laboratory, what I really will mean is both my laboratory and that of John Sidat at UCSF. We've been engaged in a long-term collaboration on this issue. That what we're interested in is looking at the three-dimensional structure and organization of chromosomes within nuclei. And this can be focused a bit, please. There are sort of two aspects to that. Imagine that this is a nucleus and these are chromosomes sort of arranged somewhat throughout the nucleus. There are two aspects to this problem. One has to do with understanding what the arrangement of these chromosomes are within the nucleus and how they behave dynamically, say as a function of cell cycle through condensation and decondensation. Asking about specific interactions between chromosomes in the nuclear envelope, or one region of a chromosome and another region of a chromosome, that sort of thing. Now that's a problem which is ideally tackleable by light microscopy. On the other hand, another aspect of our interest focuses on understanding the detailed structure of how DNA and proteins get organized into the higher order structure of chromosomes, and then how this structure changes both, again, with condensation and decondensation reactions in the cell, as well as, say, in response to transcriptional activation of a particular locus. Okay, next slide, please.
You have a remote control button.
All right. Okay, and all of these problems, of course, are really three-dimensional problems. The nucleus is very complex. Chromosome structure and organization is incredibly complex and dynamic. And there, as I said, there are two aspects to the technology for how one goes about studying these problems. One is to use three-dimensional light microscopy, which uniquely actually allows one to look in the living state in 3 dimensions at the specimens. And under ideal conditions, you can sort of push the resolution to the neighborhood of 0.1 to 0.2 microns resolution. This is limited by diffraction effects, of course, and the wavelength of the light being used. On the other hand, 3-dimensional electron microscopy, which is what I'm going to talk about here, allows one to span to very high resolutions and up to, by using high voltage microscopes, up to rather thick specimens. Okay? An interesting area, of course, is the area of overlap between these two regimes. And Lee talked a little bit about that, about looking at the same specimen in both the light and electron microscope as a way of doing correlative analysis. Okay? So that you can, in the very least, for example, one could be looking at dynamical properties in the light microscope and then transferring the information to much higher resolution in the electron microscope. The actual limitation that's listed here of approximately 50 angstroms for this sort of tomographic analysis really comes from the preservation of the specimen itself and the kind of electron doses it suffers during the analysis. This is the kinds of things that I'm talking about are being applied, or microscopy of intrinsically non-crystalline specimens. Okay, and so as Lee mentioned, that every reconstruction you do is of a unique object, and that what you then want to do is to compare sort of important features of one object to the next. You don't have the benefit of the crystallographic averaging that goes on in sort of the very high resolution diffraction type microscopy. Okay, now this is probably the most important slide. This is sort of the whole group of people that have been involved in this project in our combined labs. And as you see, and I'll try to explain why it's such an involved list, but there are really quite a number of aspects to this whole problem. But since I'm going to be concentrating on the electron microscopy issue today, I just want to highlight those people who have been involved in this. Jennifer Fung, a graduate student who is here, Michael Braunfeld, an outstanding microscopist who is also here. Craig Abbey has been highly involved in the reconstruction algorithms that we've been developing in the lab. Hans Chen and Warren Clyburn have been working on graphics and image analysis development, and by some oversight, Mei Li Wang, her name should be on here. She's also, of course, incredibly talented microscopist who many of you know, and also runs a facility at UCSF. Okay. Now, the technique for electron microscopy is a direct analog of CAT scanning techniques, which are used in medical biology to image the inside of your body. Okay. But here we use an electron beam instead of an X-ray beam to do the imaging. And so if you imagine a chromosome on a semi-thick section, say half-micron thick section, and these dots scattered around it are colloidal gold particles, much of the type that Lee talked about, which are going to be crucial for matching up the images for part of the reconstruction process. Okay? And so just as Lee talked about taking stereo pairs, okay, here what you do is you take many, many different tilted views, and we'll then try to synthesize them together in the computer to represent a single coherent three-dimensional view of what's going on inside of the object. And these colloidal gold particles are going to be crucial for the actual physical alignment of the various images, okay, with respect to one another. Okay? Mathematically, each of these tilted views can be considered as what's called a central section in Fourier space, and the idea is that you take enough views to sort of map out the whole range of Fourier space, in which case then you can do a simple Fourier transform technique to give rise to sort of internal structure in the specimen, and that's sort of the goal. Now, the actual doing of this is sort of a complicated process and broken down into what we feel are sort of three critical areas. The first area has to do with data collection, for actually collecting high-resolution tilt data sets, and I'll be telling you about approaches that we've taken to both automating this as well as to collecting very high accuracy data. Second part has to do with the tomographic reconstruction, how one actually assembles this data to generate a unique three-dimensional view of the specimen. And third, and equally important, is how you analyze and then understand what the reconstruction is telling you. Once you get to high-level specimens that are incredibly complex, this becomes non-trivial. And we've spent a considerable amount of time in our lab developing each of these three areas. Okay, so a number of these points I want to go through, just sort of an overview, and then I'll go into more detail on them. That the key to our data collection of late has been the use of a high-resolution cooled charge-coupled device to directly collect the data digitally and not have to go through the film intermediate, which we find seriously degrades the quality of the image data, as well as presents the use of digital data directly presents a tremendous savings in time for doing the reconstruction. In addition, there's things like an ultra-high angle tilt stage to try to collect as much of the tilt data as possible, and I'll go into detail on that. The other things have to do with the computer control of the microscope in an attempt to get to the point where we can fully automate the data collection process. And then we've tried to provide the sort of human interface, a completely self-contained and integrated workstation for actually dealing with all of this data and the digital image collection. Okay? And then there are other aspects of reconstruction that I'll go through. The CCD, the reason for it, as I said, is to do direct digital data acquisition. a key aspect of charge-coupled devices, which essentially act as electronic film, okay, and you sort of treat them as such. There's a shutter, effectively, that you open up. You expose it for a period of time, close the shutter, and then read it out, okay? One of the crucial aspects is that they both have exceptional linearity and incredible dynamic range. This is of fundamental importance. Can I have the lights for a second? That in the tomographic process, okay, the three-dimensional data is developed by effectively looking at the differences between a view of the image at one angle and a view of the specimen at another angle. As you'll see, to get to high resolution, what we need to do is to take many, many views, which means this difference in tilt becomes very small. And typically, we use values of around 1 to 1.25 degrees, okay? What that means is, for you to extract that information to get this level of three-dimensional resolution, you have to be able to tell the difference between an image and an image which is only tilted by 1 degree or so. And remember, the specimen is a half micron thick. So the differences are quite subtle, and so one needs a very large dynamic range so that you can detect those differences. Could I have the front lights out? Thank you. The approach that we've taken is to use a large format CCD, 1024 square pixels. This is mounted, as I'll show you. It's in the essentially where TV cameras are often mounted below the film plane in the microscope. The system is done by using a 20 micron thick single crystal YAG scintillator fiber optically coupled to a cooled CCD. Okay. The current system provides 12 bits of data, which amounts to 4,095 gray levels at a 200 kilohertz readout, and the noise is an exceptionally low value, which is due just simply to electron counting statistics. This is an outline, a schematic of the diagram of the system. This system was actually developed some years ago as a joint effort between our laboratory, Photometrics, and Gatan, and recently Gatan has now started commercializing this design on their own. The system is shown right here. This is the CCD, a fiber optic coupled to the single crystal scintillator. Now, as Ken pointed out, the actual lateral resolution that you get on your imaging effectively ends up being proportional to the thickness of your scintillator or phosphor coat, okay? And so that's why the YAG is thinned down to 20 microns to match essentially the 20 micron pixel size of the CCD, giving an optimal resolution. The rest is sort of, this stack just represents the coolers that cool the chip down, the sort of electronic coolers that cool the chip down to about minus 35 degrees to reduce dark current. Now, this whole apparatus is mounted below the microscope right under the film plane, and to facilitate use in both modes, there is a vacuum gate valve that shuts, keeping the whole CCD area under vacuum when you bring the film chamber up to room pressure to change the film. Okay, and this is what it looks like. So the microscope that we have it on is a Philips EM430. It's a 300 kilovolt IVEM. The reason we chose this machine was primarily because of the very high quality of its eccentric goniometer, which is of fundamental importance for collecting the very many views that we use in the reconstruction. Okay, so this is the CCD mounted underneath. Sort of looks more or less just like a standard TV camera mounted down there. And what we have is these are just power supply and thermal controllers for cold stages and anti-contaminators. And what there is there's a one monitor, a high-resolution monitor, which shows you the image, and another control for the computer control of all aspects of data collection. Okay. The actual system sort of schematically is configured as follows. So what we have is the microscope, the CCD. The shutter network of that is tied into the microscope so that it automatically switches the electron beam effectively to dump it into a beam stop to turn off the beam. The data is fed from the CCD system right into a computing network, which is built from a microvac system, a very high performance parallax display system, and a mercury array processor, so so that the data can be processed pretty much on the fly as it comes in. This allows you to do things like online Fourier transforms, online image enhancements, et cetera, as you collect the data. In addition, we have on our system from Philips their C400 EM controller. This sort of gives the computer control capability for the 400 series of microscopes, which is now sort of present on the newer generation computerized scopes. And the microscope then is fully under control also of the electron microscope is fully under control then of the computer system during data collection. Okay. The data collection software that we've developed is a very simple, nice-to-use menu-driven system, combined single key and command languages. This is very nice to, for example, to take a picture. All you do is raise a screen like you normally would to take a picture, and you hit G on the keyboard instead of pushing the button on the microscope. That's all it takes, and an image comes up, and now you have it directly recorded digitally, and if you want, stored on a disk. Okay, you can record up to 1024 square images. What this amounts to, and what I'm sure you'll appreciate, is that doing tomographic data collection becomes a very serious business. For example, we typically collect about 120 1024 square images. This amounts to in the neighborhood of close to 300 megabytes of data obtained at a single sitting. So the kinds of processing and analysis of all this becomes a very serious business. But computer power, of course, is becoming sort of desktop things which used to 2 years ago or 5 years ago used to fill this room. So that ends up not being a limiting thing. The display system has multiple window capabilities so that, for example, you can have on the screen, never mind, that on the screen you can be displaying, for example, the last picture you took as a reference. At the same time, you're taking this picture, so you can tell whether it needs to be translated or whether the focus is correct or these sorts of things. You can also, at the same time, in one of the windows, can automatically come up with Fourier transform for you to be checking the focus or astigmatism, et cetera. There are other aspects of image processing and sort of online and automatic correction of problems with the sort of slight variations in the gain and offset of each pixel that happen automatically. And as I say, there's also integrated control of the microscope through the C400. Now, this is, for example, a raw image collected of, this is just of a blank, can this be focused some? Of just a blank field on the microscope, okay, shown in this multi-window format on the scope. And what you see is there's slight variations. One of the problems of this particular system is that with the CCD located at the very bottom of everything in the microscope, dust and junk and sort of bits of phosphor screen and things of that sort tend to fall down and collect on that. And so you get very slight variations in what should have been a uniform field as a consequence. Okay? The slight chicken wiring pattern you see here is just a result of the fiber optic coupling. However, since the CCD is so phenomenally linear, we can completely determine what all these aspects are and correct them entirely so that none of these variations end up remaining in the image. Okay? There are also some statistical bad pixels that develop from X-ray events, and we have ways of removing those quite nicely to then ultimately give you an image which is of very high quality. Okay. The next stage is to collect very high tilt data because the limitations in resolution of the reconstruction depend not only on how many views you take, but how much of the angular space you can cover. Okay. Towards this end, we've had developed sort of again as a joint design effort between our groups and Gatan, a very high tilt stage for the Philips microscopes. And what this is an iridium tip with a very steep cutout showing where the grid is. That allows you to tilt to very high angle. And the hold-down clamps are two edge-on hold-down clamps so that there's nothing covering the central area of imaging on the grid. And this shows the back side, and they're just these two simple spring clips used to hold it down. This ends up working extraordinarily well. We can collect data. The actual place where this eclipses is about plus minus 82 degrees of tilt. Routinely, we collect data to about plus minus 75 degrees. Beyond that, we find just the thickness of the specimen gets too thick for the microscope. In addition, another aspect, once you're starting thinking about collecting such a large number of views, okay, what you do is you want to have an accurate way of reading out what angle, knowing what angle you are, and driving to the different angles. And so the system is motorized. Actually, it's an XYZ stage on the microscope plus tilt. So the normal alpha tilt here which is motorized, is under computer control, and unfortunately it's off the screen. But there's what we've done is we've put a belt here up to a very high resolution encoder, so we can get very precise readings of the actual angular position of the microscope, and that's servoed. And this is a Z drive to automatically change the Z height position. Okay. So all of this comes together to provide a very powerful integrated system for very rapidly collecting high-resolution data, tomographic data. So what used to take us, by doing it manually and developing films, what used to take us about a month and a half to end up with a data set, now we can do in an afternoon. Okay? So, I mean, it's a stunning achievement in where we've gone. And where we need to go now is to fully automate all aspects of the data collection. Right now, we still take a picture, tilt, and if you've tilted enough, you have to go in and refocus, okay, because it's not possible to make the specimen completely eccentric over such a wide angular range. However, what we are planning to do is to work out exactly from a few images, we can work out exactly what the non-eccentricity of the specimen is, and then since we have control over X, Y, and Z on the microscope, we can automatically correct for that, okay, and that's where we're headed. Okay, briefly, Zach, how much time do I have left? Okay, great, fine, thank you. The next task, okay, is to put all these images together. As I said, after sort of a day at the microscope, you'll end up with 120 images, 300 megabytes roughly of digital data. And what you have to do is to assemble that all into a coherent view. Now, as I said, in the system that we have now, as you tilt, because the specimen isn't precisely eccentric, even though the Phillips stage is very good, what you'll end up with is the image will drift somewhat, okay? And so it won't be in exactly the same position. You'll have to go track it with the normal XY positioners to recenter it in your field. As a consequence, that it's necessary for us to take the images and reshift them back so they're all precisely aligned, and also to determine precisely what the tilt axis is. Okay. Now, as you know, can I have the lights for a second? Well, as you know, as the electrons come down the column in the microscope, because there's a magnetic field there, of course, they spiral, right? And as you change the magnification on most scopes, what happens is you're essentially sampling this at different locations. So your image effectively rotates, okay? So what was a nice, what you initially knew, say this was the tilt axis that you were rotating your specimen holder about, can be some arbitrary place on your image plane by the time you're done, okay? And when you change the magnification, it'll go from being along this angle to being, say, along this angle, okay? And so what we have to do is determine exactly where that tilt plane is. As I say, we know the values from the encoder. And so, as I mentioned in the beginning, the way we do this is we put these fiducial marks on the specimen that we can then use to match up the images. And so what's required is first the recognition of the bead coordinates, okay, determination of the bead coordinates, as well as then automatically matching up which bead on which image corresponds to which bead on the next image, followed by finally a least squares procedure, which then takes all that data of all the beads from all the different images and computes then what all the orientation parameters have to be. And that was originally developed by Mike Lawrence. And one of the things we're working on then is sort of a second phase of analysis, which is much more the kinds of things that are done on the very high resolution crystallographic work, where given an initial alignment, you then do a second round of parameter refinement after you've done your first reconstruction, and then you iterate on that. What I just want to show you is an example of some of the data that we've been able for our approach for recognizing these beads automatically. Shown here is it's an image of some chromosomes and the black spots are the colloidal gold particles decorating the specimens. This is just as you would collect the data from the microscope. What we've done here is invert it to make it correspond to mass density, because it's mass density which actually is being essentially summed through the specimen by the microscope, and so the dark gold particles now show up light. And then what we've done is we've developed a filter technique which processes this image, okay, and is designed to highlight small round particles, small round objects within this image. So all the rest of the chromosomes sort of go away, and we're left with these bright spots, which then we can develop a technique which recognizes those and prunes the list based on the size of the particles, since we know what the gold beads are. What this does is it allows us to automatically recognize the gold beads, even up to when specimens are at very high tilt, where the actual amount of plastic becomes very, very thick that you're looking through. Again, that gives rise to a considerable savings in human time. The actual image reconstruction, as I said, typically these days we're using about 120 views, covering plus minus 75 degrees at 1 1/4 degree intervals. This gives us, for a specimen in the neighborhood of 0.2 to 0.3 microns thick, this gives us the possibility of having an ultimate resolution in the neighborhood of 50 to 75 angstroms. Okay, so the resolution that we will ultimately obtain is actually going to be limited really much more by our specimen preservation and preparation techniques, I think, than by the actual tomography. Now, there's a couple of different approaches, and I don't think I'll spend much time on them. I just want to mention sort of the concepts behind them. But there's a standard technique called, excuse me, filtered back projection, which is a very simple algorithm. It's actually the kind of algorithm that's used routinely in CT scanners, medical X-ray scanners. And where it's fine in medical X-ray scanners because there they collect a very, very large number of views. The equivalent number of views in a CT scan would be in the neighborhood of 500 or so views. And they cover all, where we can only go plus minus, say, 75 degrees, they can cover plus minus 90 degrees and cover all the data that exists. Okay. As a consequence, since we can't do that, there are direct consequences from having both not finally sampled the data enough as well as incompletely sampled it, which give rise to aberrations in the final reconstruction. Okay? There are now very powerful mathematical approaches that one can use to remove those aberrations in a very regular way. And there's a series of reconstruction methods that Craig Abbey in the lab has been developing based on what's called a mathematical concept called projection onto convex sets. Basically, what it does is it's a technique that allows you to provide knowledge that you know beforehand to help determine the solution. For example, what we know is that there's no such thing as negative absorbance of the electrons, basically. Everything is, all the density should be positive. There should be no negative density. The mathematics don't know that, but we, as microscopists, know that. And so we can apply that constraint. Another thing is boundary constraints. For example, we know our slab is so thick. Okay, the mathematics doesn't intrinsically know that, but by enforcing that in the mathematics of the reconstruction, we can get rid of these problems that arise from incomplete data. And then there's some other issues that one can deal with. Okay? Now, the last aspect of this, and I just want to go through this part briefly, and then I'll show you, tell you a little bit about some of the data that we have, and then end by showing you a movie of the reconstruction. Now, the hardest part is when you have, 150 million numerical values to look at from a reconstruction to decide what the hell it means. Okay, what you have is effectively a big cube of data that corresponds to the electron density or the electron scattering interactions of every point of volume within your specimen, and you have to then try to interpret that in some way to understand what the underlying biology is. Okay. And so what do we really want in a graphic system that allows us to deal with this data? What you want is something that's highly interactive, that allows you to wander through sort of three-dimensional space, sort of taking, zooming in and looking close up in areas that you're interested in and pulling back in other regions to look at a more global view, or coming in from this angle or coming in from that angle. Another thing is we want many different schemes of the kind that you heard about before for rendering the data, to ways of either looking at surfaces or looking at complete projections, whatever, as ways of helping to extract the information from that. And finally, I think crucially, is the ability to do what we call model building and quantitative analysis. Model building is what we call sort of our analog of what Lee was talking about, where one wants to follow the paths of objects or trace things in 3 dimensions within the specimen. Okay? And so what we've done is we've developed what we think is a very powerful interactive graphics system, or approach to graphics analysis, actually, for doing this. Okay. One of the things I pointed out before is this idea of multiple independent image displays, multiple windows on the screen at the same time. So you can be comparing and contrasting different things. For example, 2 windows on the screen may correspond to the left and right views having to do with the stereo pair of your specimen. Okay? It could also be, at the same time, you could have a stereo pair, and in another 2 windows on the screen, you could have, so here's a screen showing multiple views. Okay? And each of these windows are fully independent objects, okay? And you can have whatever you want in there. Now, if what is in each of these windows is geometrically a different geometric aspect of your object, for example, as I said, let's imagine this was a left view and that was a right view, okay? We could also, at the same time, in another window, be showing a perpendicular view that comes in at a different angle, okay? And then when you go in with your cursor, in 3 dimensions, as you move in this window, the cursor will move in this window and in this window simultaneously, showing you all aspects of 3-dimensional space from different viewpoints. Okay? So the problem of doing stereo pairs over a wide angular range is already been handled just by having the different images present here. Okay? Also, behind each window can be, essentially metaphorically, behind each window can be a whole stack of images. So for example, one thing that is very useful for the electron microscopy is to have showing a stereo pair of only a narrow region in the specimen. And then as you advance and back up, you move through space, moving that stereo pair forward and backwards. Okay? All of these things we feel are necessary to try to understand and be able to trace and analyze the complexities of 3-dimensional biological specimens. This just shows an example, obviously not from electron microscopy, but from light microscopy. And all I wanted to highlight here is that, again, in these independent windows, that you can be superimposing different data sets simultaneously and using the idea of different color displays as a way of superimposing that information. So if you had the same specimen stained in several different ways, you could superimpose those images on top of one another, okay, to compare and contrast the results. This, as I said, is from light microscopy showing 3 different stains. This is actual 3-dimensional light microscopy work that's done in the lab. Another way of representing the data, which we've seen before, is sort of these solid surface images. And shown here, again, is sort of looking at 2 aspects of the specimen simultaneously again, by using different colors and, in this case, transparent surfaces. Now, as I said, the key thing for us is the ability to do this 3-dimensional model building, and I just show here, this actually comes from 3-dimensional light microscopy. It's just a simpler image to display. But this is a left-right stereo pair of a 3-dimensional volume of actually looking at the arrangement of chromosomes within the nucleus, and it was very straightforward to then trace out the 3-dimensional paths of these chromosomes and mark which ones are which and put other distinguishing features on them, and to then look at them either in stereo or in movies, what have you. Okay. Now, let me sort of just wrap things up with a brief mention of sort of the kinds of things we've been trying to do in the chromosome world using EM tomography. And really what we're after is trying to understand, as I said, how DNA and proteins get organized into the unbelievably complex higher order levels of structure that occur in chromosomes, in eukaryotic chromosomes. Shown here is actually one image out of a tomographic data set from some of the earliest work that Andrew Belmont did in the lab. And this is actually a critical point dried Drosophila chromosome, which was taken in the high voltage microscope here at Berkeley. We had actually before we got the intermediate voltage microscope, we had designed and built a high tilt stage for the high voltage microscope so we could collect tomographic data there. Okay, and this just shows an example, and as you can imagine, I mean, these are incredibly complex, and to actually try to trace structures in them is quite demanding. What was clear from that early analysis was that chromosomes actually are built of a hierarchical arrangement of structures, and that the sort of propensity or the density of these structures changes depending upon the cell cycle state. And so in these embryonic chromosomes, what we saw was that in anaphase, okay, let me just go through the size classes. So the 120 angstroms is sort of the canonical DNA wrapped around nucleosome fiber, okay? That then gets built up into something which is often called a 30 nanometer fiber. In our hands, we measured it as about 240 angstroms and not 300. That just has to do with slight differences in treatment and origin of the specimen. We found a higher order size class of 400 to 500 angstroms, and yet another higher order class of about double that. And then in fully condensed mitotic chromosomes, there's even a higher order class. Now, the sizes, as I'll show you, that this class actually corresponds to something about like 1,300 angstroms in mammalian chromosomes. The point being here is that from this tomographic analysis, we're able to determine sort of the numbers, or essentially the relative populations of each of these classes as a function of condensation of the chromosomes. And what we saw was that the least condensed chromosomes, the anaphase chromosomes in for the embryonic chromosomes, had a highest population of the sort of like the 30 nanometer fiber, whereas the mitotic chromosome had the highest population of these higher order structures. Okay. The problem was we couldn't do what we wanted to do, which was actually to trace these structures, okay, within the context of the higher order structures. So the resolution wasn't sufficient doing that from these critical point dried materials. They were just too thick, and the high voltage microscope wasn't a good enough imaging tool really for doing this kind of tomography. And so we had to go to embedding our specimens in plastic sections, as I talked about, and using the intermediate voltage microscope. And this just shows you a slice through. In this case, it's now a HeLa mitotic chromosome. The gold particles decorating all over the specimen. And as you can see, this is a semi-thick section, and trying to follow details even of higher order structures in here would be quite challenging. This shows you a small region through a reconstruction of that. And one of the things I just want to point out here, not only is each of these little squiggles in here is a 30 nanometer fiber, and what we're interested in is the actual arrangement of these, but here is one of those higher order structures of 1,300 angstroms. You can see bending and looping around and then carries through into other sections. Okay. One of the things that sort of our ability to do light microscopy helped us with quite a bit was to do some simultaneous staining for both electron microscope and light microscope of the same sections. And shown here is sort of a typical image of an interphase mammalian cell. Here's the nucleus. And what one sees is a sort of typical sort of granular appearance in the nucleus, sort of heterochromatin clustered around the outside, but a rather uniform appearance throughout the nucleoplasm. And by staining this section with a dye which is specific for DNA alone, what we see is that there's a very different appearance. Okay, this is a light microscope image now, so the resolution is lower. What we see is that there's a very widely differentiated staining here. So there are regions which look quite uniform here, which are obviously not containing very much DNA. And what we see is much more of a thready appearance to all of this. And what this convinced us was some of the fundamental problems in doing imaging like this because of the lack of a DNA-specific stain for the EM. And much of what was going on here was actually being covered up by the lack of such a stain. And so what we did was we developed techniques to do very gentle extraction of the same cells, where we could compare and contrast living material using the light microscope, fixed material using the light microscope, and extracted material using the light microscope, all to verify that at least at the level of resolution of the light microscope, there had been no distortions in the chromatin. Okay? This allowed us to come up with techniques which revealed then the fact that even in the interphase nucleus, that there's a very large population of rather uniformly sized fibers running throughout the nucleus. And these are, again, these 1,300 angstrom higher order structures. And so from doing a whole series of analyses of this type, we could sort of get this very simplified model for chromosome condensation. It starts out that in S phase, one has sort of ordered and disordered regions of the chromosome. The ordered regions that you still see, which are probably heterochromatic, are about 1,300 angstroms in diameter, but they exist as sort of isolated islands, which then, as you start progressing into G2 and then into prophase, one sees that these islands start assembling together to give a rather uniform structure, which then is finally coiled into the higher order structure of the chromosome. Okay? And what our efforts have been of late have been to analyze in detail how the 30 nanometer, 300 angstrom fibers are packed within this very uniform structure of about 1,300 angstroms, which it turns out we can best isolate or best enrich in telophase. Okay? Because during telophase, what's happened is the chromosome is unwound to this level before unwinding further, and by using very simple mitotic shake-off synchronization, we can get very high populations of telophase cells. Okay. This shows such a region, and each of these things are these 300 angstrom fibers. And now I want to just end with showing a bit of this movie, and the movie will just be going through and starting with a tilt, just actually going through the tilt data set. So focus on the corner, which should be sharp. Okay. And so this is one of these 1,300 angstrom fibers, and what we're looking at is data tilted over the range of plus minus 75 degrees. Okay. And what we can see is that there are all these strands, which are the 30 nanometer fibers, all throughout that volume. And what we're interested in is tracing the three-dimensional paths of those. As you can see, this is obviously a non-trivial problem. And now this shows you walking through the 3D reconstruction in the same view that we've just had. Okay, and what you can see is these fibers twisting around and coursing through this whole volume. And we've probably picked one of the most complicated things to actually work on first. So, but this is what we've been most interested in terms of chromosome structure and biology. And this then just shows sort of an end-on view. And what you get the impression is of many strands. Okay, they're all twisting and writhing about one another as you walk forward and back. Okay, and what we believe that's going on is that, of course, the chromosome is contiguous from one end to the other, but what we're looking at is something that has a network. Actually, why don't you turn that off? And if I could have the lights. Our current model, which comes, our ideas of what's going on, which comes as a result of both Michael Braunfeld's and Karen Hahn's modeling, is that if one looks down, if here's sort of a chunk of the 1,300 angstrom fiber, which would be going off to infinity in either direction, the idea is that there are loops like this, okay, which are then super twisted like this in the chromosome. And so when we cut it at an arbitrary point and look end on, what you see is the equivalent of many strands doing this. Okay? And what we're just finding now, and we're starting to be able to trace, is these loop around points that tell us that it's coming back. And so I'll end with that and just give you an idea that the technology for this kind of tomographic reconstruction, whether it be of chromosomes, which is what we're doing, or any other aspect of things in the cells, and we've in fact even collaborated with other people on doing material science reconstructions of multi-phasic plastics that are used in things like Fierro car bodies as a way of determining domain structure in those. But that doesn't really matter. The technology and the techniques for doing this kind of reconstruction are now here, and to the point where instead of being sort of a many-month project to do such a thing, one can start thinking about doing these in a much more facile fashion, which then makes it much more accessible to the whole field, I think, of structural biology and cell biology. Thank you. Yes.
The first time I've ever seen this, the twist discussed in any detail, the image twist problem. And this problem can get very severe at low magnification. Let's say you're trying to do some metrology in a scanning microscope on something as big as an integrated circuit, you find out that things that you know are square now are parallelograms.
Well, that's actually a different issue.
It's a real problem.
I mean, the twist can work while still preserving, at least locally in the center of the image, will reasonably well preserve the geometry. The problem is, as you go to sort of wide fields, okay, the fact that the lenses are not, optical lenses are corrected for, to be what's called plan lenses, okay? These are not, and so what you see is pincushion distortions that occur towards the end of, as your field expands, you start seeing lots of pincushion distortions, which makes things which are, were supposed to be square, not anymore.
The problem here is that the coordinate system in the electron microscope is you don't have the same conditions as you have with light, and that you don't have the shortest distance between 2 points being a straight line anymore. It's a helical path.
That's right.
So you have
That's
to
right.
make a transform to a helical path coordinate system from a Cartesian
That's right.
coordinate
So
system. That's a tough piece of mathematics.
Right. Here it's not as bad because the specimen thickness is relatively small compared to the curvature of the electrons when we're looking at these high magnifications by comparison with integrated circuits. But you're right. There, where you have things on multiple levels, then you actually have to get the geometry correct. You're absolutely right.
On the movie,
Yeah.
where you're rotating the image,
Yeah.
if you use a neutral filter on one eye, the same way they did in the Super Bowl, you can see that in 3D when it's rotating one direction, and won't see it in 3D when it's rotating the other direction. Pepsi-Cola did this in the last Super Bowl. Remember the neutral filter on one eye? Or just knock one lens out of a pair of sunglasses, and you'll have a temporal separation between two views, and that'll give you your parallax angle. In your head, thinks it's looking at two different things.
I mean, often, just the simple aspect of rotating something is enough for people to get an idea that it's a three-dimensional specimen that's moving. But yes, things like that can help give you better clues. It also depends very much on which eye is dominant for you, what you want to do.
Got any way towards quantitative and measuring?
As I said, what we're in the midst of doing right now is actually working on tracing the three-dimensional paths of these fibers. And it's sort of at the early stages, and we've done enough to be able to start seeing some of these loopback regions, but we haven't done enough to be able to say that, yes, this is precisely what it is.
It's a biological question. All your chirality is saying you don't get wrong ideas.
We haven't done enough of these to tell, is the answer. Okay. No, it's a very interesting question, because there's some very interesting things with chromosome chirality, absolutely.
In that model you started to draw up there, that's the 30 nanometer fiber you're going back and forth. So how does that, where are the domain, loop domains?
The simple idea, in looking at intact chromosomes, compared to chromosomes which have been swollen by change of buffers, you don't see this idea of a fiber with loops like this, okay? You don't see this kind of central idea with loops hanging off of that. We can see that when we mistreat our chromosomes by going into inappropriate buffers, and then we can see that even in the light microscope that there's a transition from the way the chromosomes look in vivo to what's happened with these buffers. And so what we think is happening is that these things are then exploding, okay, becoming unwound and exploding. And then you end up with things that are sort of all a mass of tangles down the center, okay, and then things just hanging off because there's no longer any self-association of those bits of chromatin. And so even though the task is much harder, we've gone to really great lengths to make sure that the chromosomes remain appropriately compacted during the study. And so as a consequence, we have to go to the laborious effort of the EM tomography to then resolve what we want to see instead of blowing up the chromosome and looking at the remnants.
I wondered what induces this coiling and supercoiling?
The supercoiling we suspect is sort of just a remnant of DNA, torsional stress left over from replication. It's an idea. It's not a, we have no proof of that. We haven't done nearly enough of this in terms of looking at different points in the cell cycle to see if we can say there's more of the supercoiling in one place and less in another place in the cell cycle. some experiments that we're in the process of doing right now in the light microscope is to actually look very much at the detailed interactions of topoisomerates with chromosomes and with topoisomerase inhibitors and see if we can see aspects of this, which we should be able to see in the light microscope, and then see what the response of the sort of super twist is to the to topoisomerase inhibitors.